Good morning, everyone. I welcome you all to the CSIR online training classes. Today is the important topic discussion session. In this session, we are going to discuss about all the important topics in a different 13 units. Okay, in this 13 unit, some of the units are having more important, some of them are having less important. So, in that order, we are classified in earlier video 1, 2, 3, 4, 5, and 13 important units. So, I was given important to the topics, and I'm also including some important topics in the remaining chapters. So, we will start this session. In this session, main thing is to clarify. Okay, it's the it's a final stage of the preparation. So we want to understand the every concept and we want to recall all the concepts. So this topic is helpful you to understand the topics which are very important. They are rapidly asked. These are the topics are selected from the previous university questions. Sorry, previous year examination questions, and these topics are selected from the various websites, various expert lectures. From that I was screened these topics for your understanding. Okay, let's move to the topic. It's very easiest one. It's a very short video because this is only we are going to discuss about the in this video we are going to discuss about only the topics. Okay. Okay, this is our the my handwritten notes of this or 13 units. If you find any mistake, please ignore it. Just I am going to discuss about the topics alone, so don't find the mistakes. Take the topics and please refer it for your improvement. Okay, first unit 1. Unit 1 is only the molecular based one. Molecular structures, molecular DNA and their protein, RNA, that structure based question based thing. So in this structure based thing, we are having a first important topic is enzyme catalysis inhibition. In this, they are asking a type of enzymes, their activity, and Michaelis-Menten equations. Michaelis-Menten equation, in that we want to understand the equation and graph. It's a very short and crisp note. That is Michaelis-Menten equation. That concept, in that concept itself, you can achieve more mark. The related to the this Michaelis-Menten equation and enzymes, they are asking more questions. And then protein. In this protein primary structure and secondary structure related questions are there, and hemoglobin, myoglobin are related questions, oxygen saturation curve. This is very important in this very important topics in the unit 1. And apart from that, basic things, pathways. Okay, these are all the pathways in your biochemical Leninger book, or your the biochemistry peoples. You know, these are all the important thing in our life metabolites, life metabolisms. Okay, glycolysis is very important topic. TCA cycle, Ph.D, electron transport chain, gluconeogenesis, pentose phosphate pathway, and glycogen metabolism, and role of insulin and epinephrine in the pathways. Okay, what is the things? And then common introduction about the bioenergetics. And next to this first portion. Okay, that is very important. And then some important topics in unit 1 is the inhibitions of various pathways. What are the inhibitors are available, which inhibitor inhibit which protein, which enzyme. That is the thing very important. Then biosynthesis process is normal one, and the Switterian related concept, that is the pKa value calculations, and classification of amino acids, collagen, elastin role, and very important, that is the Ramachandran plot. Okay, very important. Okay, this topic is very important in a unit 1. And protein folding, denaturation compounds are very important. Denaturation, which protein this is the problematic related question. This kind of question they provided the temperature or denaturation factor, which using that compound we want to understand the question and give the analytical based question. And protein interaction, that DNA microarray related questions. And what is the types of DNA structures are available and their role. And likewise the RNA structure and function. Biosynthesis of how the RNA and DNA synthesis, that is the biosynthesis of the RNA and DNA is very important. The pathway is very important. Next, in the first unit, the next portion is a purine pyrimidine. This purine pyrimidine structure based question is very important. Okay, the purine pyrimidine synthesis pathway and structure based question is very important. And next to it is the van der Waals force related, that is the interaction between proteins, amino acids in a protein. And pK value calculation, this is very important. There are many online videos are available for pK pi value, so please refer that. This pK pi value is very very important in this unit 1. Okay, next is sequencing techniques based on the molarity. They ask the problems how to calculate molarity, normality, that is the mole and normal based questions are there. Okay, and pH buffer preparation, del G calculation, epimers, anomers, glycosides bonds, which role of glycoside bonds, that is the thing very important in this unit 1. They are asking problematic question like the problematic and analytical question with this glycosidic bonds. And glycosaminoglycans, that is very important topic in the latest questions. And lipids and membranes, beta oxidations, fatty acids, and a different type of lipids. This very 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 important in the unit 1. Okay, next is the unit 2. In this unit 2 they are asking a nearly 30 to 25 mark. Okay, that nearly we can expect a 12 to 13 question. Okay, first thing is what are all the membrane compounds and their roles. It's very very important. There are n number of compounds available in the membrane. We want to know the roles. Okay, next is the phospholipids, that and their role. In this unit, the diffusion by osmotic process, that membrane transport process is very important. And next is RBC membrane. Then active transport and passive transport is very important. Which are the compounds which involved in the element involved in this active transport is called as the element. That is very also important. Okay, next is a lipid raft. This lipid raft is also very important. And freeze etching, and the main important thing is the pumps, channels, transporters are involved in membrane transduction. The very very important topics. We want to know the name and their function. This is we can expect the match the following related question. Okay, next is the membrane potential, that the problematic portion. Actin, dynein, kinesin, these are all the very important very important topics in this unit. Once you understand the concept is enough, because they are asking a problem analytical question, so you just need to understand what is this, what is the role of this. That the basic is very important. And role of Golgi complex and mitochondria, peroxisome and lysosome, these are all the organelles. Okay, cellular organelles. This organelles playing a vital role in many functions in a cell. So that we want to find out the role. Next thing is a disease caused by the deficiency of enzyme. Okay, which deficiency of the enzyme? Some of the enzymes lack. If the enzymes lacked in the human body, so we may receive many diseases. That is the thing here we want to find out. Okay, first thing is the we want to know which are the enzymes responsible for the diseases. Then we want to understand what are all the diseases may come. This is also the match the following related question. This very important topic. In this CSIR examination, we may expect many match the following questions. Among them, it is expected one. So just to study this concept with examples. Okay, disease and enzymes, which all the enzymes are responsible for the diseases. This is also very important. Next is the heterochromatin, euchromatin that transposes chromosomal structure function. This is the next one is DNA sequencing. This DNA sequencing is a very important topic. Sanger sequencing and next generation sequencing. That is the things are very important. Then centromeric region in acetylation methylation packing, it's playing a vital role because DNA acetylation methylation, this process, which enzymes are involved, which is what is the process level, that is very important. Cell cycle, in that we have checkpoints. In that checkpoints, the CDK, CDC and P21, P53, what is the role of these compounds, and we want to understand what is the important compounds present in this pathway, cell cycle phases. Okay, next is microbial growth kinetics. We can expect problematic question in that part, in this genre. Okay, next is the microbial disease and the microbial names, it's also very important. Okay, first another so once again I'm tell the disease and the enzyme, this is one concept. Okay, disease and the enzyme is one important thing. And the microbial disease and the microbe name, this is the match the following question, it's very important. Okay, next is say stress response and cancer stages. This cancer, it's totally important. Okay, cancer initial stage, metastasis, apoptosis, all those things are very important. Okay, next to the unit 2, the is unit 3, that we can expect 22-25 mark, nearly, that is we can expect 9 to 10 questions. That is fundamental process of the unit. In that unit, major thing is the enzymes involved in the initiation, elongation, termination process of replication, transcription. We want to understand eukaryotic and prokaryotic. The enzymes are very important. Just go through any PPTs or any notes, just to understand what is the enzymes involved. Okay, next is the topo isomerase telomere replication. This is a most repeated question in the previous year questions, and this experimental based questions are also very important. Okay, next is helicase activity, functions of ribosome enzyme and semi-conservative model. This pattern of inheritance of heavy and light chains, these are all somewhat they are asked in a previous year question. And this experiments related to the conservative and the conservative and dispersive, this concept is very important. And Wakasagi fragment, and what is the Wakasagi fragments, Skillner fragments. This is the understandable question in part B. We can expect this question in the part B. The polymerase 3, DNA polymerase 3, is a very very important topic in a CSIR examination. And then RNA modification step is very important step in every molecular mechanism. So RNA modification is very important. That is uh, also asked in they are repeatedly asked in the previous year question. Then polyadenation, capping and editing, silencing technique is the latest one. It's also important in this important topics. The DNA damage and repair pathways, we want to study the MMR, NER, BER and DBR method. These are all the pathways which involve in the DNA, and the types of DNA, and what is the enzymes cause this DNA the mutation. Okay, this is very important. What is the type of mutation, the cause which is involved in that pathway, this is a match the following related question. And DNA repair method and the damage DNA, what kind of damage and what is the repair method, that is also match the following damage and the repair method. Okay, this is the thing in the batch of the following related question. And homologous combination is very important. And then site direction recombination, DNA splicing, spliceosome compounds which are, and very 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 important topic is the CRISPR-Cas. This is very very important. Okay, this CRISPR-Cas is the most repeatedly asked question in the last 2 year questions. Okay, another is a lacopron and tryptophan concept and RNAi. Then protein synthesis, it's enzymes. What are the proteins? What are the enzymes are involved in the protein synthesis? Which protein is synthesized by which enzyme? That is also very important. Gene regulation, that we want to know, lytic and lysogenic pathways. RNA polymerase role and function, structure also very important. These are all about the unit 3 important questions. And next one is a unit 4, very 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 important topic. Okay, in this unit 4, we can expect nearly 13 to 14 questions. In this unit, we may achieve 30 to 35 mark. First one, we want to know what are the bacterial disease, what are the viral disease, yeah, what are all the airborne disease, what are the waterborne disease, which pathogen involved in that disease, mode of attack, and causative agent for the disease, and what is the host for the disease. This is the very important topic. We can expect at least two questions in the part C. So it's very important. And pathway based one. Okay, in that pathway one based one, GPCR, trypsin kinase, MAP kinase, JAK-STAT, WNT, Hedgehog, TGF, beta, and NF-kappa B, Notch pathway, afferin, and beta catenin pathway. These are all the cat pathways are very important, because this is all the very biggest one. How can we study this? For this kind of question, just refer many PPTs are available in the internet. Just refer the PPTs to understand one concept and then move to the next topic, because in the CSIR examination you want to understand the role, function, the basic concept of the particular topic. That examination, they are asking a story, they are telling the story. From that story itself, they are asking a question. So if you know the concept, you can easily solve the story. Okay, next, apart from the pathways, the next thing is the secondary messengers. Secondary messengers, cyclic AMP, GMP, calcium, IP3, and the NO, nitrogen oxides. Okay, these are the role and their functions in the pathways. That's very important. Apart from that, cell communication, this that cell communicates, cell communicate, how it is communicate, that cadherin, selectin, endocrine, and immunoglobulin role in that cell communication. Okay, next topic is very important. Topic is apoptosis. Apoptosis pathway is very important, and related to apoptosis, we can expect nearly 2 to 3 questions in an exam paper. Okay, next is a hallmark of the cancer is very important. As I mentioned in the earlier chapter, that cancer pathways are very important. That's these P53, 21, P16, and PRB. These are all the very important elements in that topic. And how that normal cell is converted to the cancer cell, that is also a very important topic in the 4th unit. And what are the cancer name? Okay, breast cancer, other lepidopterin, that is the drug name. Okay, what is the drug name? How the drug will inhibit which protein? Okay, so that is a match the following with that question: cancer name and the drug name. Okay, lung cancer, we can use this drug. Okay, that is very important thing in this unit 4. And oncogenes are oncogenes and their role, tumor suppressor genes, and angiogenesis and metastasis process. This is a common topic. Just to understand what are the proteins are involved, what are the pathways are involved. Next thing is immunology. Immunology, we have innate this is a vast topic. In that we want to study the specific kinds. What is innate immunity, adaptive immunity, what are the molecules involved, what is the pathways involved. Then told like receptor concept, monoclonal antibody production and application, MHC molecule, vaccine production, and vaccine, which vaccines helpful for the which disease. Okay, that is very we can expect the match the following: vaccine name, pathogen name, and disease name. That is the match the following related question. Captain, adjuvant, that is the adjuvant based research questions we can expect. Hypersensitivity and their types, cytokines and their roles. Cytokines CD4, 8, and 27 are very important. Okay, and the last is the cell matrix. What adhesion adhesion functions and adhesion molecules related questions are there. Okay, unit 5, that is developmental biology. In that we can expect nearly 10 to 12 question we can achieve 20 to 25 question, 25 mark. Okay, first thing is very important topic, okay, because the unit 5 and 4 is very important. This is very, we can focusly study some topic and we can achieve more marks, so that's why only that is important. Okay, get development and stages and concepts are very important. So organogenesis, and we want to study some important terms. Okay, as I mentioned earlier, the CSIR examination is analytical based. We can expect more analytical question. So we want to understand the concept first, and then only we can move. So first we want to know the terms. What is potency, commitment, one specifications and their types. Okay, specification we have three types. So we are, so that types: induction, competence, cytoplasmic, determinant, and differentiation. What is determinant and differentiation, and morphogenic gradients? What, these are all the basic terms. We want to understand what is it and what is the where it is playing a role. Okay, next is the fertilization process. In this process, what is the process, receptors, which what are the enzymes and receptors involved, polyspermy, what is the polymerase stage, and chromosomal number. These are all the process. For sea urchin, we want to know, we want to study an amphibian, mammals, and capsitations are very important. This is most asked terms, most asked question in a CSIR previous year question. This concept is very important. Just Google it with the, okay, sea urchin fertilization process, amphibian fertilization process, mammalian fertilization process, and capacitation fertilization process, and study the concept and stages. These are all the, you just want to just read the concept and you can easily achieve the good mark. And types of eggs and cleavage patterns. These are all the very much cleavage pattern of birds, insects, amphibians, and mammals. And valve formation in a C. elegans, and the axis formation in the sea urchin is a very important concept in the previous year question on CSIR unit 5. Gene involved in a gastrulation. Gastrulation is a very very important topic. Okay, most repeatedly asked question is a gastrulation related question. The frag, sea urchin, gastrulation, C. elegans, and C. brevis, and the sea urchins are very important. Chick related questions are very important. These are only the genes involved in the gastrulation. Okay, what are all genes means? So that is the hunchback, caudal, and noggin, beta catein. These are already some genes which involved in the gastrulation process. Just study this. Gastrulation is very very important in this unit 5. Okay, next to this second part of the unit 5 is the eye lens induction. Eye lens induction of frog, invertebrates. Okay, vertebrates and invertebrates both, and plant. Okay, this till now we discussed the animal for animal developmental process. Here we want to study the plant development process. In that very important concept is the ABC model. ABC model of the Arabidopsis thaliana and Adrenum is very important, and Drosophila development process is very important. Apart from that, the Drosophila development is very important. It is specifically we want to study this topic. And limb formation of Hox gene, which the Hox gene, HER and FGF. These are all very important. And CAMP signaling, it's very important. Dichosterium, which playing a vital role in this development process. That study is very important. Plant, how the leaf develop, how the flower develop, and how the meristem is developed. These are all very important. And embryogenesis of chick is very important topic in this unit 5. Next is unit 6 is very very easiest. We can achieve more mark with less study. Okay, photosynthesis pathways, C4, C3 CAM pathway is very important, and also Kelvin pathway. Okay, apart from this, the second thing is the metabolite production. First one is pathway, second one is the metabolite production. How the metabolites are produced, under which condition it can produce. And plant hormones is very very important. Only 5 hormones and you can achieve a nearly 10 mark. Okay, so if you well versed about this 5 hormones, you can nearly achieve 10 marks easily. So there's 5 hormones. You can how it is synthesized, how it is transported, how it is involved in the signaling. Nitrogen metabolism. Okay, very very important. This nitrogen metabolism. What are the microbes involved in this step? And phytochrome. What is the types and functions? Okay, next is a callus plant tissue culture. Very important things: callus development, shoot root development. What are all the compounds involved in the plant callus development and shoot root development? And Rubisco Kelvin pathway is very important thing, as I mentioned earlier. And bacteria is one nitrogen, and secondary metabolites pathway is very important. And cryptochrome and phototropin, short day plant and long day plants. This is all very important. Short day plant and long day plant is very important. Among that, the biological clock and totipotent is very important. And animal physiology, we can expect this is a vast unit, so we want to study selectively. So endocrine is a which are the what are the hormones involved, or the hormones are produced from the which gland. That is we can expect the match the following related question. And what are the amino acid precursors? Now, this is the endocrine system. In the respiratory, we can title the volume, respiratory volume. We want to title and IRV and ERV. And then the gaseous exchange in the process, and the myoglobin and hemoglobin concept. And next to that, cardiovascular system, ECG, that ECG waves related question is very important. This ECG waves related question is very important in this unit. And excretory system, what are the ions concentration in that excretory system, and what are the essential elements which involve in that is in the excretory system. The urine production, okay, that is very important, the formation of urine, and role of nephrons in that okay process. And the vitamin deficient and the disorder is very important. And we can expect the match the following related question. What are the vitamins are there, what is the disorder, the deficiency make this which disorder. These are only concept, okay, in the unit 7. Next is the unit 8. This unit is a vast one. We want to study the selective concepts alone. Okay, unit 7, 8, 9, 10, 11. Okay, unit 6, 7, 8, 9, 11, 8, 9, 10, 11, 12. These are all the topics are we want to that is a very vast syllabus, so we want to selectively study the particular concept. Okay, genetics. In that, genetics, Mendelian genetics and non-Mendelian genetics are genetic related concept we want to understand. Molecular biology of the prokaryotic and eukaryotic is very important. We are studied in a third unit itself, so no need to study in this here. Previous units itself, that unit 6, unit 4, unit 3 itself, we are studied the concept of molecular genetics about prokaryotic and eukaryotic. And here also very important things is the terms, okay, very important terms, that is the codominant, incomplete codominant, and pleiotrophy, the pedigree diagram. These all the things are very important. HFR mapping, genetic math related problems are there, chromosomal mapping, mutagenesis, ABO blood grouping model, mitotic meiotics, chromosomal alterations, and phylogenetic inheritance QTC model. These are all the important in this unit. And unit 9 is a diversity based question, which is very very important, which is very very easiest unit in this among the 13 unit. This is very easiest unit, because we are going to study the selective manner and we can achieve more mark. Phylogenetic tree, what is the interaction, what is the relation, cladogram based question. This is very important topic in the previous year questions. And the reproduction system, what is the reproduction principle of molecular, fungi, plant, animal, which are all the which are all the hormones involved in that, what are the hormones are involved in that is a very important thing. And alpha beta gamma taxonomy is very important. Causative organisms, the disease and host. This is we can expect the match the following. Rare endangered species list and where it is placed, okay. For example, you can expect the lion tail monkey, that is only present in the India and Tamil Nadu. So that is a rare endangered species and their places. And conservative strategies, what is the conservative strategies they used, okay. Next is the properties of the phylum, classes, and hotspots of animals. This three is very important. We are going to study. We can spend at least to study this ninth unit. We just two to 3 hours is enough to study. You can achieve more. But just study for the your understanding. Next is the ecology. Ecology question. This is somewhat vast question for biotechnology people, but for your zoology and some people, it's the easiest one. Habitats and niche, hydrosera, xerosera types, succession. So types of successions. The rules is very important. Okay, the rule is very important. Rules based questions are there. Population ecology. In that population ecology, recapture the recapture method and types of depressions are very important. And population growth correlated that problems are we can expect population related problems, metapopulation, types of survivorships curve, R and K selection. This is a Cardi Wingberg equation is law is very important. We can expect this concept. Okay, repeatedly they are asking this concept, RK selection and Hardy Weinberg law. Next is a community interaction. In this is the ecology have a big the very important thing. So amenity interaction, community interaction, symbiosis. What are the microbes are involved in the symbiosis? Commensalism, parasitism, and competition, paradition. Okay, this what is the definition and the example is very important. Next thing is the model. Okay, this model is the vodka model is also important. That data list, the data list is very important in this unit. Greenhouse effect, COD POD related problems also they are asked. List of project tiger zones. Tiger zone is very important because it's an Indian project. We can expect more question. National parks and their the habitat, the animal or bird. An ecosystem. In this ecosystem, the energy cycle, primary secondary production of energy in this ecosystem, and food web and food chains, global warming, and the lentic ecosystem is very also important, and types of ecosystem and their locations are very important. Okay, next is 11th unit is evolution. In this evolution also we are going to study the selective topics. Okay, as I mentioned, unit 6, 7, 8, 9, 10, 11, 12. These are all the we want to study selective topics. Okay, that types of selection, directional, stabilized, and descriptive. These are the selections are very important. The founder effect is very important. The battle like these are all the experiments. Okay, evolutionary study, these are all the experiments, landmark concept, Darwin's concept. These are all the concept. This concept based question, the basic experiments are very important. Aerobic, anaerobic metabolism, the concept of operin, the Holden, it's also the experiment. The experiment of Miller, genetic drift related experiments are very important. Biological clock, genetical clock is very important. Time scale is very important. And reciprocal alteration and different types of organs. Organs means that homologous, analogous organs and their examples. Types of evolution is and concept of speciations are very important. Applied in this unit 12, you can expect more questions related to the research. Okay, the very repeatedly asked question is related to the agrobacterium. Agrobacterium related is mediated gene transfer is very important. Agrobacterium application, role, their characters, function, all those things are very important. As I mentioned, the types of vaccine, what is the type, what is the vaccine is helpful for which disease, and vaccine development process is very important. Here also the tissue development, the animal tissue culture and plant tissue culture, which are all the chemicals conditions you can use in the this animal and plant tissue culture is very important. Root shoot culture and development, and types of fermentation is very important in this unit as well. Microbes and their production, which microbe produce which product. the microbe and the food microbes also used in the food production. For example, the curd production that is used for, we can use the lactobacillus. Likewise, they are asking many product there and their microbes. It's a match the following related question. This is very very important topic. Okay, this is very very important topic, types of fermentation and their microbes and their products. Then sometimes they ask which extracellular product or intracellular product, so that microbes and the product is very important. Somatic embryogenesis, psychotic embryogenesis, marker genes, TI plasmid. This TI plasmid is very 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 important person in this unit. Plant genes delivery system and gene therapy. Gene therapy, now currently available gene therapies and their application. Vector based vectors. Okay, vectors is very important. Cosmates, plasmates, very important in this vector based study. And biosensors, their application and the field. Okay, how it is activating biosensors. They have different type. How it is activating, what are the application in the medical field, that is very important. Fire remediation, what are the microbes used for metal degradation, dye degradation, and plastic degradation, timber waste degradation, all those things are there. So fire remediations, we want to study with the microbes. Next is very 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 important topic is the unit 13. This unit 13 we can expect nearly 25 to 35 mark. That is means that uh, 13 to 14 questions. That very very important topic in the unit 13 is all the topics are very important. Among that, recombinations technology is very important. Recombinant technology, what are the enzymes they used, what are the vectors they used, what is the role, structure, function, all the nook and corner of the recombinant technology is very important. And then methods, 2D gel electrophoresis, 2D 1D gel electrophoresis, hydroelectric focusing, and hydroelectric isoelectric focusing gel preparation. That is a very important thing. Casmate, bacterial artificial chromosome, yeast artificial chromosome concept is very important. Whole genome library preparation, cDNA library preparation, ECG, PET, MRI, fMRI, CAT, brain record. These are all the instrument based question. Next, PCR is very important. PCR. This is very, 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 very important question. Nested PCR, hosted PCR, colony PCR, real time PCR. Apart from that, the RFLP, RAPD, AFLP, microarray. These are all the very important topics, and mutagenesis, deduction, and gene knockout. This, as I mentioned earlier, the unit 13 is very important for the problem and analytical question. Part C, we can expect more questions from the unit 13. Part C, it's a very important for this unit 13. Okay. Next is the protein sequencing. Sequencing is technique, Sanger sequencing, next generation sequencing, Lumina, entire and gene microchips based questions. And instruments like NMR, use of NMR, use of X-ray, FTR, CD. CD means circular dichromatism. Here we want to study the instrument setup. You want to study the method, how it will work, and where it will help. That is the thing. Okay, NMR is helpful for protein structure prediction. That is the one keyword I'm telling. NMR extra FTR, the instrument application. ELISA, okay, this is immunological techniques. ELISA, radio immuno assays. Okay, Western blotting, FACS, flow cytometric analysis, fish dish, and double division method. These are all methods for immunological based question. Microscopical technique, and in that microscopic technique we want to analysis the some term light microscopy. What is the nanometers for the images, what is the clarity, what is the aperture. These are very important. And staining method, different staining method, bacterial, gram back, positive, negative, polysaccharide containing microbe, and staining techniques is very important. This all about unit 13. And as I mentioned, we are discussed most important topics of CSIR Life Science syllabus. Okay, this topics are most repeatedly asked in the previous year questions, and this topics are very easiest topics. You for your short term period preparation, you can make the for example Sankar sequencing technique means Sankar sequencing technique PPT. Just Google it and view the PPTs and refer the concept if you have a short time period. In that same way, if you have a 6 months period, if you have a 1 year period, means you can study with the books. Okay, go for a book, go for a material, just read out the concept and you can easily get a better idea about the Sanger sequencing technique. Okay, as I mentioned in the previous videos, the unit 4 5 13. Okay, okay, uh, part C we can expect more questions from 4 5 13. Okay, and apart from that, the part C, part B having 1 2 3 and I think so 6 and 12. Okay, these 8 units are very important than the other units. Why I mention means these topics are very easiest and focused topics in CSIR exam preparation. Okay, so only they mention. The remaining topic is we can expect that is unit 7, 8, 9, 10, 11. These 5 units are also important, but we will study with specific selective topics. Okay, this was very important content of this video. Thank you. Just study these topics and go with the confident, because this are most repeatedly asked like questions, so you can easily solve the CSIR paper. Okay, and upcoming days I'm having a plan. I don't know how it is possible. As soon as possible I will try to make a video for unit 1 and upload upload in this channel, because that is in a progress. Unit 1 is worst one. I was not completely finished the unit 1, so only I'm waiting for uploading. So if I am finished unit 1 with a better manner, I will upload the video. Otherwise you will study these concepts and attend the CSIR examination. Okay, I hope I will meet